Welcome to the Deep Dive, the show where we unpack complex ideas and extract the most important insights from your sources. Today, we're taking a journey into the hidden world of plant engineering, right beneath our feet and soaring high above our heads.
Indeed. Our mission in this deep dive is to explore how plants, despite being rooted in place, manage to acquire and transport all the essential resources they need: water, minerals, carbon dioxide, and light. It's really an incredible story of ingenious adaptations that allowed plants to colonize land and thrive.
And we're kicking off with an image you might have seen that seems a bit mysterious. Have you ever wondered why aspen leaves quake and shimmer even on a day with, very little wind? It's this fantastic light display, right? A shimmer of radiance dappling the forest floor. You'd think they're just being dramatic, wouldn't you?
Yeah, but what's truly fascinating here is that the mechanism is built right into the leaf's structure. The petiole, that's the slender stalk connecting the leaf to the stem, it's flattened along its sides. This unique design allows the leaf to flutter almost exclusively in the horizontal plane.
Okay, so why does that constant trembling matter? I mean, is it just for show?
Well, here's where it gets really interesting. The leading hypothesis is that this quivering actually increases the photosynthetic productivity of the entire plant
The whole plant. How?
by allowing transient shafts of sunlight to reach those lower shaded leaves. The aspen is essentially optimizing its energy capture, making sure no photon goes to waste. It's a surprising adaptation, isn't it?
Yeah, it really is. That tiny flutter isn't just pretty. It's a brilliant, almost imperceptible design choice that dramatically boosts the tree's energy production.
Exactly. It's a testament to the powerful evolutionary pressures that have shaped plants into the masters of resource acquisition they are today. We'll be looking at the evolutionary journey plants took to get here, their incredibly sophisticated transport systems, how they pull essential nutrients from the soil, and the powerful forces driving water movement.
We'll also explore the incredible partnerships plants form with other organisms, how they regulate water loss, and the elegant flow of sugars throughout their bodies. Think of this as your shortcut to understanding the silent, incredible biology happening all around you. Okay, let's unpack this evolutionary journey then. Our sources reveal that the algal ancestors of plants had it, well, pretty easy, just soaking up water, minerals, and CO2 directly from their aquatic environment. Every cell was bathed in everything it needed. Simple.
Right. But colonizing land presented huge challenges. Suddenly, plants faced intense competition for light, water, nutrients. Everything became scarce, relatively speaking. This drove the evolution of critical features.
Like what?
Well, early non-vascular land plants, like mosses, developed waxy cuticles and fewer stomata to prevent excessive water loss, but they were still limited in reach. They couldn't get very big.
So, with these early plants facing such limitations, what was the game-changing evolutionary step that allowed them to truly expand their reach?
That would be the evolution of vascular tissue xylem and phloem. This was the big leap, the internal highway system, if you will. It allowed for extensive root and shoot systems, paving the way for long-distance transport.
Right, the plumbing.
Exactly. Xylem carries water and minerals from roots to shoots, while phloem moves sugars, the products of photosynthesis, from where they're made or stored to where they're needed.
So once that internal plumbing was in place, natural selection really went to work, shaping shoot architecture for efficient light and CO2 capture.
Precisely. Stem strategies became incredibly diverse. You have tall plants developing thick stems to avoid shading. You have vines relying on other structures. There's an energy trade-off, between growing tall and extensive branching.
And the leaves themselves.
Leaves, of course, were a major innovation. Broad surfaces maximize light capture, and those stomatal pores allow for CO2 diffusion. But it's always a compromise with water loss.
Always a trade-off.
Always. What's fascinating is how leaves are arranged. Research shows the ascending spiral arrangement of leaves, often at a specific angle, 137.5 degrees, minimizes self-shading. It ensures each leaf gets its share of sunlight.
That specific angle, wow.
Yeah, and when lower leaves become unproductive, the plant even practices self-pruning and sheds them. It cuts its losses.
Smart. And below ground, roots aren't just sitting there, are they?
Oh, not at all. Roots are anything but passive. Research shows plants can rapidly adjust their root architecture and physiology. If they encounter a patch of low nitrate, for example, they tend to extend straight through it.
Okay.
But if they find a pocket rich in nitrate, they branch extensively, and they synthesize more transport proteins to absorb those nutrients efficiently. They really respond to their environment.
That makes sense.
And what's truly intriguing is how plants might distinguish self from non-self to reduce root competition.
How would they do that?
Well, for example, buffalo grass stolons develop fewer and shorter roots when near cuttings from the same plant compared to those from a different plant. It raises the question, how does a plant know? We're still uncovering those answers.
Fascinating.
And this leads us perfectly into the idea of crucial mutualistic relationships, like those with mycorrhizae, which we now think were a critical step, maybe the critical step, in land colonization.
Okay, so once these resources are acquired, they need to move around inside the plant. And you mentioned these intricate systems for both short and long distance transport. Let's start short distance. Within cells or between adjacent cells?
Right. For short distance movement, the plasma membrane's selective permeability is absolutely key. It controls what goes in and out of each cell.
And this is where plants are a bit different from us animals, right?
Yeah, fundamentally different in some ways. In plants, hydrogen ions, H plus B, play the primary role in establishing what's called the membrane potential and driving active transport. It's all about pumping protons.
Instead of sodium, like in our nerve cells.
Exactly. In animal cells, sodium ions, Na plus O, are typically involved, but plants use this proton gradient. For example, an H plus sucrose cotransporter uses the energy stored in that H plus gradient to actively pull sugar into cells against its concentration gradient.
Okay, so they pump protons out, then let them back in, bringing sugar along for the ride.
That's the basic idea, yes. Plants even have specialized ion channels, like the potassium channels in guard cells that control the stomata, those pores in the leaves. Think of them as tiny, very specific gates.
Got it. And for moving things between cells within a tissue, you mentioned two pathways.
Yes, the apoplast and the symplast. Think of the apoplast as the plant's intercellular hallways and outer surfaces, all the cell walls, the extracellular spaces, even the dead cells like mature xylem vessels. It's everything outside the plasma membranes.
Okay, the outside network.
Right. The symplast, on the other hand, is the connected network of all the living rooms, the cytoplasm or cytosol of all the living cells, linked together by tiny channels called plasmodesmata that go through the cell walls.
So the inside network, all connected.
Exactly. And substances can move via three main routes. The apoplastic route, just along the cell walls, the symplastic route moving from cytoplasm to cytoplasm through plasmodesmata, or the transmembrane route, where they cross plasma membranes repeatedly, moving from cell to cell.
And can they switch between routes?
Oh, yes, absolutely. Substances often use a combination of routes to get where they need to go. It's quite flexible.
Okay, so that's short distance. What about water movement specifically? How do we predict where it will go?
For that, we need to understand water potential, symbolized by the Greek letter psi-s. It's simply the physical property that predicts the direction of water movement. Water always moves from regions of higher water potential to regions of lower water potential.
Water flowing downhill.
Exactly like that. It's moving down an energy gradient. The equation looks like S plus P.
Okay, break that down.
AS is solute potential, sometimes called osmotic potential. It reflects how much solutes, like sugars or salts, tie up water molecules, making them less free to move. Pure water has a solute potential of 0. Adding any solutes makes AS negative. The more solutes, the more negative it gets.
So solutes lower the water's energy or potential. And AP.
AP is pressure potential. That's the physical pressure on the water. This can be positive, like the turgor pressure inside a plant cell where the protoplast pushes against the cell wall. That's what keeps plants stiff and prevents wilting.
Right, turgor pressure.
Or pressure potential can be negative, like the tension or pull found in xylem tissue when water is being pulled up the plant.
Okay, positive pressure pushes, negative pressure pulls.
You got it. So if you place a flaccid or limp plant cell in pure water, which has high water potential, water rushes in because the cell's potential is lower due to its solutes. The cell swells and becomes turgid.
It stiffens up.
Exactly. But if you place that same cell in a solution with a higher solute concentration than the cell itself, water will move out of the cell towards the lower water potential outside. The cell undergoes plasmolysis. The plasma membrane pulls away from the cell wall.
It shrinks inside its wall. Ouch.
Pretty much. And speeding up this water movement across membranes are aquaporins, specialized protein channels just for water. They facilitate rapid water transport.
Water tunnels, basically.
Good analogy. And there's a fascinating insight from a scientific exercise mentioned in the source, looking at radish seed water uptake. Its Q10 value, that's a measure of how much a process speeds up with a 10 degrees C temperature increase, was close to 1.
Meaning temperature didn't affect it much.
Right. which suggests that the initial soaking up of water by sites is primarily a physical process driven purely by these water potential gradients rather than an active metabolic process, even though it looks like something a living thing is doing.
Interesting. So physics, not just biology initially.
Precisely.
Wow.
Now, for long-distance transport, diffusion and active transport across membranes are way too slow to get water from roots to leaves or sugars from leaves to roots. This is where bulk flow comes in.
Bulk flow. What's different about that?
Unlike osmosis or diffusion, which depend on concentration gradients across membranes, bulk flow is the movement of the entire solution due to a pressure gradient. It's much faster, it moves everything together, water and solutes, and it's independent of solute concentration, at least directly.
Like a river flowing, not just molecules diffusing.
Exactly. And the structure of xylem and phloem facilitates this. The absence or reduction of cytoplasm and organelles in the mature, conducting cells, the xylem vessels and sieve tubes makes them like hollow pipes, minimizing resistance to flow, like an unclogged drain, as the source puts it.
Makes sense. So all these mechanisms, membrane transport, apoplast, symplast, water potential, bulk flow, they all work together.
They orchestrate the movement of resources throughout the entire plant, from the smallest root hair to the tallest treetop.
Okay, let's switch gears slightly. What are plants actually made of? What nutrients do they need from the soil?
Well, if you look at a plant's dry mass, take out the water, most of it, about
Okay.
96% is carbon, oxygen, and hydrogen. That comes from CO2 in the air and water via photosynthesis.
Right, the basics.
But that remaining 4% is crucial. It's made up of inorganic minerals absorbed from the soil. And we call an element essential if it's absolutely required for a plant to complete its life cycle, grow, reproduce, the whole deal.
And how do scientists figure out which ones are essential?
Mostly through hydroponic culture. That's growing plants in carefully controlled mineral solutions without soil. You can omit one element at a time and see if the plant suffers.
Clever. So what are the big ones?
We distinguish between macronutrients, needed in relatively large amounts, and micronutrients needed in tiny quantities. The main macronutrients are carbon, oxygen, hydrogen, of course, plus nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium.
NPK, or the big three in fertilizer, right?
Exactly. Nitrogen, phosphorus, and potassium. They're vital components of organic compounds, proteins, nucleic acids, chlorophyll. Nitrogen, especially, is often the most limiting nutrient for plant growth worldwide. Plants need a lot of it.
And the micronutrients.
Yeah.
Needed in tiny amounts.
Right. Things like chlorine, iron, manganese, boron, zinc, copper, nickel, molybdenum, sometimes sodium. They typically function as cofactors, helpers for enzymes. Iron, for instance, is critical in the cytochromes involved in electron transport during respiration and photosynthesis.
So small amounts, but still essential for key reactions.
Absolutely. And diagnosing deficiencies can be like plant forensics. The symptoms often give clues. For instance, magnesium deficiency causes chlorosis yellowing leaves because magnesium is a component of chlorophyll.
Makes sense.
And it also depends on whether the nutrient is mobile within the plant. If it's mobile, like nitrogen, potassium, or magnesium, the plant can move it from older leaves to younger growing tissues. So deficiency symptoms show up in the older leaves first.
Okay, the plant sacrifices the old for the new.
Pretty much. But if the nutrient is immobile, like iron or calcium, it stays put once it's incorporated. So deficiency symptoms appear first in the young, developing leaves because they can't steal it from older parts.
Okay, so the location of the yellowing tells you something.
Exactly. For example, in corn, nitrogen deficiency causes that classic yellowing, starting at the tip of the older leaves, moving down the middle. Phosphorus deficiency gives younger leaves a reddish-purple tint. Potassium deficiency causes drying along the tips and edges of older leaves. Very distinct patterns.
And I read a curious fact in the source. Zinc deficiency in fruit trees can sometimes be cured just by hammering zinc nails into the trunk.
Yes, it sounds crude, but it can work. It allows the zinc to slowly leach into the tree's vascular system. Not the most precise method, but sometimes effective.
Amazing.
Huh.
Now, what about managing the soil itself? This has a long history, right?
Oh, huge history. Ancient farmers, realizing that adding manure or other amendments fertilization could maintain soil fertility, was likely a key factor in the shift from nomadic life to permanent settlements. Soil became a renewable resource, enabling agriculture and specialized labor.
And today, we have commercial fertilizers.
Right. Modern fertilizers usually supply nitrogen N, phosphorus P, and potassium K. They're often labeled with an NPK ratio, like 15-10-5, meaning 15% nitrogen, 10% phosphate, P2O5, and 5% potash, K2O.
What's the difference between those and organic fertilizers like manure or compost?
The main difference is release rate and composition. Commercial fertilizers offer minerals in a form that's immediately available to plants, but they can be easily leached by rain, potentially causing runoff pollution like algal blooms in lakes. Organic fertilizers release nutrients more slowly as they decompose, which also improves soil structure. So, trade-offs.
In soil pH, the acidity or alkalinity matters too.
Critically, soil pH affects how available different minerals are to plants. At a high pH, say around 8, plants can absorb calcium easily, but iron becomes almost unavailable because it precipitates out. At low pH, other minerals might become too soluble, even toxic.
Like aluminum.
Exactly. Low soil pH is a major global problem, especially in the tropics. It causes toxic aluminum ions to become soluble, which stunts root growth and severely limits crop yields. It's a huge issue for food security in many regions.
So soil isn't just, well, dirt. It's a complex system.
Absolutely. It's a living ecosystem. Topsoil, the upper layer, is a complex mix of mineral particles, sand, silt, and clay, plus living organisms and decaying organic matter called humus.
And the particle size matters.
Yes, the texture, the relative amounts of sand, silt, and clay affects water retention, aeration, and drainage. Sandy soils drain quickly and don't hold water well. Clay soils hold lots of water but can get waterlogged and lack air spaces. Loams, which have a good mix of all three, are generally the best for agriculture.
And the chemistry, you mentioned negative charges.
Right. Most soil particles, especially clay and humus, have negatively charged surfaces. This is important because they attract and hold on to positively charged ions or cations nutrients like potassium K+, calcium Ca2+, magnesium Mg2+.
So they don't get washed away easily.
Exactly. Negatively charged nutrients, anions like nitrate, NO3, or sulfate, SO4, aren't held tightly and are more easily leached from the soil by rainwater.
So how do plants get those positifications off the soil particles?
Through cation exchange, plant roots actively pump out hydrogen ions, H+. These H+ ions displace the nutrient cations bound to the soil particles, releasing them into the soil solution where the roots can absorb them.
They trade an H+ for a K+ or Ca2+?
Basically, yes. And humus, that decomposed organic matter, is incredibly important. It prevents clay particles from clumping, it helps retain water, provides air spaces for roots, increases the cation exchange capacity, and serves as a reservoir of nutrients that are released gradually as it decomposes.
And it feeds the soil life.
Absolutely, and the amount of life is staggering. A single teaspoon of topsoil can contain about 5 billion bacteria, plus fungi, algae, protists, insects, earthworms, nematodes, plant roots. It's a bustling underground city. Earthworms, for example, are hugely beneficial. They mix the soil, create channels for air and water, and their castings enrich the soil.
Okay, so speaking of soil life, you mentioned plants aren't just taking from the soil, but there are partnerships. Mutualism.
Yes, exactly. It's not just exploitation, it's often a two-way relationship. Mutualism, where both partners benefit, is incredibly common and vital across all kingdoms of life.
Any examples beyond plants?
Oh, loads. The source mentions pufferfish getting toxins from bacteria they host, leafcutter ants farming specific fungi, lichens being a partnership between a fungus and an alga or cyanobacterium, ants defending plants that provide them nectar. It's everywhere.
Wow. Okay, back to plants and soil. What about bacterial helpers?
Right. There are specific bacteria that are hugely beneficial. We categorize them broadly as rhizobacteria, which live in the rhizosphere, that's the soil layer immediately surrounding the plant roots and endophytes, which actually live inside plant tissues, between or within cells.
And what do they do for the plant?
Both types thrive on nutrients secreted by the plant roots, like sugars and amino acids. In return, they can do amazing things. Stimulate plant growth by producing hormones, produce antibiotics that protect roots from disease, absorb toxic metals, or make nutrients more available. For instance, some bacteria can increase phosphate solubility.
So they're like external gut microbes for the plant.
That's a pretty good analogy, actually. And cutting-edge research using metagenomics, studying the collective genetic material from a whole community, is revealing incredibly complex and diverse bacterial communities associated with roots. This might lead to new agricultural strategies, maybe inoculating crops with beneficial bacterial cocktails.
Very cool. Now, the nitrogen cycle. You mentioned nitrogen is often limiting. Why is it so tricky?
Because even though the atmosphere is about 79% nitrogen gas, N2, that N2 molecule has a super strong triple bond between the two nitrogen atoms. It's incredibly stable and inert. Plants simply can't break that bond and use N2 directly.
So they need it in a different form.
Exactly. They primarily absorb nitrogen as either ammonium ions, NH4+, or nitrate ions, NO3. And getting atmospheric N2 into those forms relies almost entirely on bacteria.
Okay, so what are the steps?
Well, first there's ammonification, where decomposers convert the nitrogen in dead organic matter back into ammonium, NH4+. Then, crucially, there's nitrogen fixation. This is certain bacteria converting atmospheric N2 into ammonia, NH3, which picks up a proton in the soil to become ammonium, NH4+. This is energetically very expensive, requires something like 16 ATT molecules for every 2 ammonia molecules produced.
Wow, that's costly.
It is. Then other bacteria perform nitrification, converting ammonium first to nitrate, NO2, and then to nitrate, NO3. Plants can absorb both ammonium and nitrate, though nitrate is often more readily absorbed.
But isn't there a step where nitrogen is lost?
Yes, unfortunately. Denitrification is carried out by other bacteria, especially in anaerobic, waterlogged soils. They convert nitrate back into N2 gas, which escapes into the atmosphere. That's a loss of usable nitrogen from the soil ecosystem.
Okay, so it's a complex bacterial balancing act. What about that famous legume partnership?
The symbiosis between legumes like peas, beans, clover, alfalfa, and rhizobium bacteria. This is the most efficient system for nitrogen fixation in agriculture. The bacteria induce the plant roots to form swellings called nodules.
I've seen those on clover roots.
Inside those nodules, the rhizobium bacteria transform into a specialized form called bacteroids. These are the nitrogen-fixing powerhouses.
And how does it work inside the nodule?
What's truly amazing is that the plant cells within the nodule produce a protein called leukhemoglobin. It's an iron-containing protein very similar to the hemoglobin in our blood.
Leukhemoglobin, like legume hemoglobin.
Exactly. Its job is to bind oxygen very tightly. The enzyme nitrogenase, which fixes nitrogen, is poisoned by oxygen. So leukhemoglobin acts as an oxygen buffer, keeping the free oxygen concentration extremely low, creating the anaerobic environment needed for nitrogen fixation.
But the bacteria still need oxygen for their own respiration, right, to make all that ATP.
Precisely. Leukhemoglobin delivers just enough bound oxygen to the bacteria for their respiration without letting free oxygen interfere with nitrogenase. It's an incredibly elegant solution.
That is amazing. And the exchange.
The plant provides the bacteria with carbohydrates produced during photosynthesis. In return, the bacteria fix atmospheric nitrogen into ammonia, which the plant can then use to synthesize amino acids and other organic nitrogen compounds, transporting them up to the shoot. It's a classic mutualism.
A perfect partnership. What about the fungal friends, mycorrhizae? You mentioned them earlier.
Right, mycorrhizae. These are incredibly widespread mutualistic associations between plant roots and fungi. They might be even more important overall than the rhizobium symbiosis because they occur in so many more plant species.
And they were crucial for colonizing land.
Yes, the fossil record suggests mycorrhizal associations were present in the very earliest land plants, 400–500 million years ago. It's thought this partnership was essential for both the plants and the fungi to succeed in the harsh terrestrial environment.
So what's the deal? What's the exchange?
Similar principle. The plant provides the fungus with sugars. The fungus, through its extensive network of microscopic filaments called hyphae, which make up the mycelium, dramatically increases the surface area for absorption.
So the fungus acts like an extension of the root system.
A massive extension. This greatly enhances the plant's ability to absorb water and, crucially, mineral nutrients, especially phosphorus, which is often immobile and hard for roots to get. The fungi can also secrete growth factors that stimulate root growth and antibiotics that protect against pathogens.
Wow. Are there different types?
Yes, 2 main types. Ectomycorrhizae form a dense sheath or mantle of fungal hyphae over the surface of the root. The hyphae also extend into the soil and grow between the cells of the root cortex, forming a network for nutrient exchange in the apoplast. These are common in woody plants like pines and oaks.
Okay, mostly on the outside.
Right. Then there are arbuscular mycorrhizae, sometimes called endomycorrhizae. These are much more common, found in about 85% of all plant species, including most crops. They don't form a dense mantle. Instead, their hyphae penetrate through the root cell walls, but then invaginate or push into the plasma membrane without actually breaking it.
Like pushing a finger into a balloon.
Exactly that analogy. Inside the cell membrane, they form these highly branched structures called arbuscules. They look like little trees. This is where the nutrient exchange happens, providing a huge surface area right inside the root cell, functionally speaking.
That's incredibly intimate.
It really is.
Yeah.
And it has practical impacts. Treating seeds with fungal spores can significantly boost crop yields, especially in nutrient-poor soils. Conversely, invasive species like garlic mustard can sometimes succeed by disrupting the native mycorrhizal networks that other plants depend on.
Fascinating complexity. Are there plants that get nutrients in even weirder ways?
Oh, absolutely. Beyond mutualism, you have epiphytes, plants like staghorn ferns or many orchids that grow on other plants, but they're not parasitic. They just use the host for support and absorb water and nutrients from rain and dust.
Okay, aerial plants.
Then you have parasitic plants like dodder or mistletoe. They actually tap into the vascular system of their host plant to steal water, minerals, and sometimes even sugars. Some, like dodder, lack chlorophyll entirely and are totally dependent on their host.
Plant vampires.
Pretty much. And finally, you have carnivorous plants, pitcher plants, sundews, Venus flytraps. These are usually photosynthetic, making their own sugars, but they live in nutrient-poor environments, typically acidic bogs low in nitrogen. So they've evolved these incredible mechanisms to trap and digest insects to supplement their mineral nutrition, especially nitrogen.
So they eat bugs for the nitrogen. Nature finds a way, doesn't it?
It certainly does.
Okay, so we've covered acquiring resources, these amazing partnerships. Now let's get back to the grand transport systems.
Xylem and phloem, moving stuff over long distances. You mentioned the scale, an average tree moving 800 liters of water a day. That's huge.
It is immense, and it all starts with root absorption. Root tips, and especially the root hairs growing out from them, provide a massive surface area for taking up water and dissolved minerals from the soil solution.
And they first enter the cell walls.
Right, water and minerals initially enter the hydrophilic walls of the epidermal cells, moving along the apoplast pathway between the cells.
But then you mentioned a gatekeeper, the Casparian strip. Exactly. As the water and minerals move inwards through the root cortex via the apoplast, they eventually reach the endodermis, the innermost layer of cells in the cortex surrounding the vascular cylinder, where the xylem and phloem are located.
Okay.
The cell walls of these endodermal cells have a special band of waxy, waterproof material called the Casparian strip. It's like waterproof caulking between the cells. This strip completely blocks the apoplastic pathway.
So water can't just seep between the cells anymore.
Correct. It forces all water and minerals that were traveling the apoplast to cross the selectively permeable plasma membrane of an endodermal cell before they can enter the vascular cylinder. Anything already in the symplast just continues through the plasmodesmata.
Ah, so it's a mandatory checkpoint. Ensures the plant controls exactly what gets into the xylem highway.
Precisely. It ensures selective uptake of minerals and prevents water and solutes from leaking back out of the xylem into the cortex.
Okay, so once inside the vascular cylinder, the xylem sap water and minerals starts moving up. You said it's pulled, not pushed.
Yes, primarily pulled. This upward movement is by bulk flow, and the driving force is transpiration, the evaporation of water vapor from the surfaces of cells inside the leaves, which then diffuses out through the stomata into the atmosphere.
So evaporation from the leaves pulls water all the way from the roots. How does that work?
This is explained by the cohesion-tension hypothesis. It relies on the unique properties of water.
Okay, break it down.
Step 1. Transpiration. Water evaporates from the moist surfaces of the mesophyll cells lining the air spaces within the leaf. This happens because the air inside the leaf usually has a lower water potential, is drier, than the cell surfaces. Water vapor then diffuses out of the leaf through the stomata, down its concentration gradient into the typically even drier air outside.
Okay, water leaves as vapor.
Step 2.
Oh.
Creating tension. As water evaporates from the cell wall surfaces, the remaining water film retreats into tiny pores in the cell wall. Surface tension causes this air-water interface to curve inward, creating a negative pressure potential tension. Think of it like sipping liquid through a very narrow straw.
A pulling force.
Exactly. Step 3. Cohesion and adhesion transmit the pull. This negative pressure, or tension, pulls water from the more hydrated parts of the leaf. Because water molecules are strongly attracted to each other through hydrogen bonds, that's cohesion, they form continuous, unbroken columns in the xylem.
Like a chain of water molecules.
Precisely. And water molecules are also attracted to the hydrophilic walls of the xylem vessels and tracheids. That's adhesion. So the tension generated by evaporation in the leaves is transmitted all the way down these water collars in the xylem, through the stem, down to the roots, and even out into the soil water.
Wow, so the sun evaporating water from the leaves is ultimately what pulls water up hundreds of feet.
That's the essence of it. The energy for this massive water transport is ultimately provided by the sun driving evaporation. It's solar-powered bulk flow. And think back to that quivering aspen leaf we started with. That movement likely enhances airflow around the leaf surface, boosting that crucial transpiration pull. It's all connected.
Incredible. But with all that leaf surface area needed for photosynthesis, plants must lose a ton of water this way.
They do. It's a major dilemma. Maximizing CO2 uptake inevitably maximizes water loss. About 95% of the water a plant absorbs is lost through transpiration, mostly via the stomata, even though stomata typically cover only 1–2% of the leaf's surface area.
So those tiny pores are critical control points. How do they work?
Each stoma, or pore, is flanked by a pair of specialized guard cells. These cells control the diameter of the stoma by changing their shape.
How do they change shape?
By changing their turgor pressure. When guard cells actively pump potassium ions, K+, into themselves, this decreases their internal water potential, makes it more negative. Water then rushes in by osmosis, causing the guard cells to swell and become turgid.
Okay, K plus N, water falls, cell swells.
And because of the unique orientation of cellulose microfibrils in their cell walls, when they swell, they bow outward, increasing the gap, the stoma between them.
So swelling opens the pore. How do they close it?
Just the reverse. Potassium ions leave the guard cells. Water follows by osmosis, the cells become flaccid, less bowed, and the gap between them closes.
What triggers them to open or close?
Several cues. Light is a major one. Specific blue light receptors trigger K+ uptake and opening. Also, low levels of CO2 inside the leaf air spaces, which happens when photosynthesis starts using it up, can stimulate opening. And plants have an internal clock, a circadian rhythm, that causes stomata to open during the day and close at night, even if kept in constant darkness.
They anticipate sunrise.
Seems like it. But environmental stress overrides these. During drought, roots and leaves produce a hormone called abscisic acid, or ABA. ABA travels to the guard cells and signals them to close the stomata, conserving water even if light and CO2 levels would normally cause opening. It's a crucial survival mechanism.
A trade-off again, save water, but reduce photosynthesis.
Always a trade-off. And don't forget, transpiration also provides evaporative cooling. It can lower the leaf temperature by as much as 10 degrees C compared to the surrounding air, which can be vital in hot, sunny conditions to prevent enzymes from denaturing.
Right. And desert plants have special tricks.
Absolutely. Xerophytes, plants adapted to arid conditions, have numerous adaptations. Some just have very short life cycles timed with rainy seasons. Others, like cacti, have highly reduced leaves, spines, and fleshy stems that store water and carry out photosynthesis. And many use CAM photosynthesis, crassulation acid metabolism. These plants, like succulents and pineapples, open their stomata and take in CO2 at night when it's cooler and more humid, minimizing water loss. They store the CO2 temporarily as organic acids. Then, during the day, they close their stomata tightly to conserve water and release the stored CO2 internally to be used in photosynthesis, powered by sunlight.
Clever. Taking CO2 at night, doing photosynthesis by day with the door shut.
Exactly. Okay, finally, let's talk sugar transport, phloem translocation.
Right, moving the sugars made the leaves to everywhere else. How is phloem sap different from xylem sap?
Very different. Xylem sap is mostly water and minerals, quite dilute. Phloem sap is thick and syrupy. Its main solute is sucrose table sugar, often at concentrations as high as 30% by weight. It also transports amino acids, hormones, and other signaling molecules.
And it moves differently too, not just upwards.
Correct. Phloem sap moves from a sugar source to a sugar sink. A source is any part of the plant that is a net producer of sugar, usually mature leaves, actively photosynthesizing. Or it could be a storage organ, like a tuber or root, that's breaking down stored starch to export sugar, say, in the spring.
Okay, producers or storage releasing. And sinks.
A sink is any part of the plant that is a net consumer or depository of sugar. This includes growing roots, buds, stems, fruits, or it could be storage organs that are actively stockpiling starch for later use, like roots or fruits accumulating reserves in the summer.
So the direction can change depending on the season or the plant's needs.
Absolutely. A storage root can be a sink in the summer and a source in the spring. And interestingly, adjacent sieve tubes within the same vascular bundle can actually be transporting sap in opposite directions simultaneously, depending on where their connected sources and sinks are.
Whoa. How does the sugar get into the phloem at the source?
That's phloem loading. Sugar, usually sucrose, moves from the mesophyll cells where it's made into the sieve tube elements and their associated companion cells.
Boy.
This often requires active transport because the sucrose concentration inside the phloem is usually much higher than in the surrounding cells.
So it needs energy to pump sugar in.
Often, yes. It typically uses that same chemiosmotic mechanism we talked about earlier.
Hmm.
Proton pumps create an H plus gradient, and then H plus sucrose cotransporters use that gradient to move sucrose into the companion cells and sieve tube elements.
Okay, active loading. Then what happens at the sink?
Phloem unloading. Sucrose diffuses out of the phloem into the sink tissues, where the concentration is lower because the sugar is being rapidly consumed for growth or respiration, or converted into insoluble starch for storage. Water then follows the sugar out by osmosis.
So how does this loading and unloading drive the flow?
This leads to the pressure flow hypothesis, the main model for phloem transport. Sugar loading at the source makes the phloem sap there very concentrated, lowering its water potential. Water rushes in from the adjacent xylem by osmosis, building up high positive hydrostatic pressure, turgor pressure at the source end.
Okay, high pressure at the source.
At the sink end, sugar is unloaded, making the phloem sap more dilute. Water leaves the phloem by osmosis, lowering the hydrostatic pressure at the sink end.
Low pressure at the sink.
Exactly. This difference in pressure, high at the source, low at the sink, creates a pressure gradient that drives the bulk flow of the entire phloem sap solution from source to sink, like water flowing through a pipe from a high pressure point to a low pressure point.
So xylem is pulled by negative tension. Phloem is pushed by positive pressure.
That's a great way to summarize it. Xylem flow is driven by transpiration and negative pressure, while phloem flow is driven by active sugar loading, unloading, and positive pressure.
And this source-sink relationship has practical implications.
It does. Sometimes a plant produces more flowers or young fruits than it can support with the available sugar from its sources. In this case, it might undergo self-thinning, naturally aborting some flowers or fruits so the remaining ones can develop properly.
The plant prioritizes.
Exactly. And growers use this principle. They might manually remove some flowers or young fruits from, say, an apple tree. This reduces the number of sinks competing for sugar, resulting in fewer but larger and often sweeter apples at harvest.
Makes perfect sense when you understand the flow. Wow, what an incredible journey through the inner workings of plants. Resource acquisition, nutrition, transport. Let's quickly recap our main takeaways from this deep dive.
Yeah, absolutely. First, I think it's clear that plants are truly masters of resource acquisition. They've evolved specialized roots for mining the soil and incredibly diverse leaf and stem structures perfectly adapted for capturing light and CO2.
hmm And their internal transport systems, the xylem and phloem, are just incredibly efficient highways, driven by pressure gradients and the unique physical properties of water itself.
Right, and we learn that soil isn't just inert dirt, it's a dynamic, living ecosystem. Plants depend heavily on it, often through these fascinating mutualistic relationships with bacteria and fungi, especially for getting vital nutrients like nitrogen and phosphorus.
Those partnerships, like the rhizobium nodules and mycorrhizae, are just amazing examples of coevolution.
They really are. And finally, plants show these remarkable adaptations to constantly balance the need for photosynthesis with the need to conserve water. Think of stomatal control and CAM plants. It's a clear testament to evolution shaping elegant solutions to environmental challenges.
Yeah, that continuous silent movement of water and sugars throughout the plant, powered by sun and pressure. It really is an elegant example of nature's engineering.
Couldn't agree more.
So think about the next time you see a towering tree, or maybe just a delicate wildflower. Behind its stillness lies this astonishing dynamic network of transport and exchange, a whole hidden world of pressures, potentials, and microscopic partnerships. What other everyday biological mysteries are waiting for your deep dives?
We certainly hope this deep dive has given you a newfound appreciation for the incredible biology happening all around us and inside plants. Until next time, keep exploring, keep questioning, and keep learning. This has been The Deep Dive.